My dear students, welcome to EPG Pathshala. I am Dr. P. Suganya, working in the Department of Food Processing and Preservation Technology, Avinashlingam University. Louis Pasteur demonstrated that the microorganisms is responsible for fermentation. In 18th century, Nicolas Appert proved successful preservation of foods in glass container is nothing but the sterilization process, followed by tin-coated steel containers were used. At present, a combined heat treatment of UHT and aseptic processing provides good quality foods without affecting nutritional qualities in cost-effective way. In today's module, we will look into ultra-high temperature process and aseptic processing in detail. Let us see the objectives of this module. After going through this session, you will be able to understand ultra-high temperature process and to understand the aseptic processing and packaging. Pasteurization. During pasteurization, the liquid foods are treated at 72 degrees Celsius for 15 seconds, where denaturation of protein, modification in amino acids, rancidity of fatty acids, destruction of vitamins and reduced minerals like calcium, phosphorus and trace minerals occurs. These biochemical changes takes place along with the destruction of microorganisms. Pasteurization process failed to inactivate bacterial spores. Since bacterial spores have very good heat resistance due to its thick protective outer layer, these spores are resistant to even drying, freezing and preservatives. To inactivate these spores and to retain the nutritional qualities, ultra-high temperature process is required. The ultra-high temperature processed liquid products are further processed by aseptic processing method. Aseptic processing is a unique technology where the product and packaging materials are sterilized separately and food materials are filled in the hot condition and packed in the aseptic conditions. If you go back to conventional canning system, food materials are pre-treated to inactivate the enzymes. Then food material is filled in cans in hot condition to avoid oxidative damage. Required headspace is essential to avoid problems during retorting. The can is hermetically sealed, followed by retorting at optimum time, temperature and pressure. Most of the times, flexible retort pouches are also used in the sterilization process. In such cases, the packaging material must withstand up to 100 to 110 degrees Celsius for high acid foods and 125 degrees Celsius for low acid foods. Since low acid foods contain heat resistant bacteria and microbial spores which needs to little high temperature to inactivate the microorganisms and spores. Though canning and retorting are very successful technology with commercial sterility, nutritional content and other properties. The tin plate containers are heavy in weight which leads to rusting also. The cost involvement is high. Now let's see about ultra high temperature processing. Continuous flow of heat at a temperature of 135 degrees Celsius for very short time of 2 to 5 seconds destruct almost all the microorganisms and spores present in the food. This is known as ultra high temperature processing. Residential time varies with respect to the product. So few of the ultra high temperature processed foods are the liquid foods such as milk, fruit juices, cream, etc. and foods with solid particles like baby foods, tomato products and vegetable soaps and larger particles like stews. Now coming to the method of UHT processing. Two basic methods of UHT processing are direct heating and indirect heating. In direct heating, the high temperature steam is applied directly to the food material under strictly controlled conditions. Processing temperature of 140 degrees Celsius is quickly reached and hold it for 4 seconds. The temperature is reduced by flash cooling in a vacuum vessel. The short time process will not affect the nutritional qualities. Two different types of processing under direct heating are injection and infusion. First one is injection. The high pressure steam is applied into the food product by steam injector which raises the temperature rapidly. Then the product is flash cooled in a vacuum condition. since this method is energy intensive and cause flavor damage. This method is suitable for limited food products. The second one is infusion. The product is pumped through a distributing nozzle into a chamber of steam with high pressure. More surface area of the product is exposed to steam in this system. Immediately after processing, flash cooling applied to the product in vacuum chamber. Infusion method is instant heating and cooling process. The localized overheating is not applicable and highly suitable for low and high viscous products. And next one is the electrical heating method. The electric current is directly passing through the product. This system is applicable for liquid foods and particulate foods. The electrical resistance for liquid food and solid food creates problem in this system. The second one is indirect heating. A partition placed between the product where the partition acts as a heating or cooling medium. The heating medium passes through one side of partition and food products pass through the other side of the partition through the partition plate where heat is transferred. The similar process occurs during cooling also. In this method, there is no direct contact between the product and heating medium. Flow rate of the product, physical properties of the food material, residential time and cleaning are the important parameters to be considered before processing. Three types of heat exchangers such as plate heat exchanger, tubular heat exchanger and scratch surface heat exchangers are used. The plate heat exchanger is used for low viscous liquid products. Tubular heat exchangers are mainly applicable for liquid foods with particulate foods with specific size, and scratch surface heat exchangers is highly suitable for viscous food material with or without particulate foods. Let's see the advantages of ultra high temperature processing. The application of very high temperature for minimum process time leads to retention of nutritional qualities. UHT processed products has longer shelf life up to 6 months without refrigeration. UHT processing is independent of packaging size and cost of the packaging material is low. Now, let's see about aseptic processing. The term aseptic is coined from a Greek word septakos, which means the absence of spoilage microorganisms. The filling of commercially sterile product into a sterile container under sterile condition is known as aseptic processing. These products are self-stable at room temperature for long duration. Now, we see about the development of aseptic packaging. In 1914, development of sterile filters for wine industry was developed. In 1915, patent granted for pure pack cotton, that is folded blank wax cardboard package, was introduced. In 1948, first commercial successful equipment was introduced. In 1950, the three important aspects like aseptic canning system and UHT processing of cream and Tetra pack introduced tetrahedron package and primarily for milk and liquid dairy products. And next in 1963, Tetra pack initiated using rectangular cotton Tetra brick. In 1981, FDA approved hydrogen peroxide as sterilization agent for polyethylene contact with food product. And Ocean Spray Company introduced juice boxes to the market. In 1985, packaging juice drinks in PET bottle was introduced. And 1990, advances in higher speed filling equipments for high density polyethylene and polyethylene terephthalate packaging were introduced. In 2003, aseptic filling of non-carbonated beverages in cans in France was introduced. And now let's see about saline features of aseptic processing. Increased shelf life of product at room temperature is highly possible for aseptically processed foods. Low cost and lightweight materials such as paper, flexible packages and semi-rigid materials which are not applicable for other sterilization process can be used, and space requirement also very less. The ultra high temperature process for aseptically packaging is highly efficient and thermally suitable, which leads to high quality product with comparable with high nutritive value with pasteurized foods. The two types of aseptic applications followed in food processing technology are: the first one is pre-sterile and sterile foods. Packaging of food materials such as milk and dairy products, fruit juices, soups and liquid foods with particulate foods are pre-sterilized and steriled products. Second one is non-sterile foods. The packaging of food materials like fermented dairy products such as cheese, yogurt are non-sterile products. Aseptic processing and packaging is a complex process because sterilization in 3 different steps is very crucial to achieve successful aseptic processing. First one is sterilization of equipment such as ultra high temperature section, gas and air in the line should be properly sterile. The second one is sterilization of food materials before packaging. The third one is sterilization of packaging materials and closure before filling. First, let's see about product sterilization. The thermal bacteriology is important to distract target microorganisms and spores. The time temperature combination for the treatment to inactivate spores decides the commercial stability of the product. The pre-sterilization is desired where the product is treated with UHT processing. Proper thermal treatment and subsequent rapid cooling applied to foods where required viscosity is important to avoid problems during filling. The rapid heating and cooling process retain most of the nutritional qualities of foods. The heat exchanger system used for sterilization is already discussed in ultra-high temperature processing. The second one is sterilization of packaging materials and equipments. High temperature steam, chemicals and radiations are used for sterilization of equipments and packaging materials. These agents are used alone or in combination with each other may be applicable. Limited sterilants are used in aseptic system. The first sterilant is heat. The product supply line and filling system are sterilized by moist heating method. A high temperature steam under pressure or hot water is used here. Also, superheated steam or hot air may also be used for sterilization equipments. Time temperature requirement is high for dry heat method when comparing to moist heat methods. To assure appropriate sterilization in aseptic processing and filling machineries, high temperature and holding time is required since the equipment is made of stainless steel. In moist heating method, temperature ranges between 121 degrees Celsius and 129 degrees Celsius is used, whereas in the dry heating method 176 degrees Celsius to 232 degrees Celsius is employed. The temperature range between 260 to 350 degrees Celsius is applied for sterilization of air by incineration. The second sterilant is chemicals. In general, various acids, ethylene oxide and ethanol are used for primary usage, but hydrogen peroxide is considered as effective sterilant for sterilization of aseptic processing unit. At room temperature, hydrogen peroxide is not an effective sporicide. Hence, hydrogen peroxide at 30 to 35% concentration is used as sterilant in most of the packaging materials. Then, hot air at 60 to 125 degrees Celsius is used to dissipate the residual hydrogen peroxide in the packaging material. The third sterilant is radiation. In aseptic system, to decontaminate packaging materials, gamma radiations are used due to its penetrating power. Packaging materials are treated in bulk in commercial irradiators. 1.5 mega rad is usually applicable for sterilizing the container or packaging materials for acidified foods. Aseptic filling of low acid foods are also possible where the dosage required for sterilization is high. Low acid foods are easily liable to microorganisms and hence higher dosage is required for sterilization. Ultraviolet C light is an alternate. Due to its less penetration capacity, UV light is used only for food contact surfaces. Next one is the filling of product. The product to be sterilized has been brought to the holding tube. This holding tube provides required residential time at particular processing temperature. Though some loss of heat occurs during the flow of product, the required product temperature for sterilization is maintained. No external heat is applied to the holding tube. In most of the aseptically processed products, de-aerators is used to remove air prior to packaging. The de-aerators are used to avoid oxidative reactions which leads to defects in quality of the product. The de-aerator consists of vessel where the product is exposed to continuous vacuum. The sterilized product is collected in an aseptic flow tank before packaging. The end of the cooling section is connected with valve system at one end and connected with packaging section at the other end. The product is pumped into the flow tank and is removed by a positive pressure in a tank with sterile air. This pressure is controlled to protect it from contamination. The container must be filled without contamination by organisms either from the equipment surface or from the atmosphere that surrounds the filter. The filling is done in an enclosed area that is supplied with sterile atmosphere. The next one is the seals and closures. The maintenance of sterility and integrity of the closure and seals during handling and transportation is most important one in aseptically processed foods. Mainly to avoid recontamination, the importance is given to seals and closures. The longitudinal and transverse seam or the system manufactured in Tetra Pak. In the longitudinal system, a flat web of packaging material is used, supplied in reels. This flat material web is formed into a tube which is sealed longitudinally, resulting in a cylinder-shaped structure. The strength of this longitudinal seam is determined partly by an overlap seal and partly by plastic longitudinal strip. This strip is first sealed in one edge of the packaging material web, and once the packaging material tube has been formed, sealed to the inner surface of the packaging material. Both these operations, the strip application and the actual longitudinal sealing, are done by using sterile, hot air and pressure. Transverse sealing is done below packaging material tube. By constantly moving sealing and pressure jars, the pressure is applied from the outside of the packaging material tube, squeezing the product from the sealing area. An electrical impulse is passed through the sealing jar and the heat is transferred from the outside of the inside plastic layer of the packaging material. The polyethylene layer is heated, melted and pressed together between the pair of jars. While pressure is maintained, the melted plastic layer cools down and bonding is brought between the two opposite packaging material surface. Aseptic packaging lines. The 5 basic types of filling and sealing line in aseptic processing are: the first one is containers made of glass, metal, thermoformed plastic or filled in sterile environment and sealed. This is called fill and seal method. Second one is the roll of flexible material is sterilized and packed is formed in aseptic condition and sealed. This is called form, fill and seal method. Example, Tetra Pak. Third one is by using knockdown blank, erected, sterilized, filled and sealed of packaging is called as erect, fill and seal method. The fourth one is rolls are sterilized, thermoformed, filled and sealed in the sterile conditions. For example, plastic soup cans. The last method is blow mold, fill and seal of product in the sterile environment. Now let's see about the requirement for aseptic packaging materials. The packaging material should well match with the food material to be packed and should have barrier properties. To maintain the sterility and physical integrity is important. The packaging material should withstand sterilization methods. The material should protect food from light, oxygen and to retain aroma and flavor. Now we look into aseptic packaging types. Numerous consumer packaging are available in the market. First one is carton boxes. Aseptic carton boxes are used for particulate food packaging. The second one is bags and pouches. For packaging of milk, pillow pouches are used. This type of pouches are used in aseptic processing for particulate foods with particle size of 12 micron and bag size from 1 to 5 liters. The third one is cups and trays. Pre-made or formed, filled and sealed in, that is FFS, that is form, fill, seal machine. Usually polypropylene based multi-layer materials with ethyl vinyl alcohol material is used for this purpose. Next one is the bottles and jars. Food containing small particles like baby foods are aseptically packed in glass bottles. Jars are applicable to larger size particles of around 12 mm cube. Recently, returnable pouches are used for ultra high temperature treated milk. Closing is done by heat sealing aluminum lids. The next one is metal cans. Steel and aluminum cans are used for aseptic filling. The next category is plastic cans. Filling and closing of two-piece cans, example, gourmet cans was developed. Cans and lids have easily opening features which are made of polypropylene and ethyl vinyl alcohol. These materials are sterilized with UV radiation and hydrogen peroxide. These type of cans used only for packaging of coffee. The next one is composite cans. This type of cans used for fruit juices with long fibers. Next, we will see about the familiar packaging type of Tetra Pak aseptic cartons. The carton type Tetra Pak made of three basic materials such as polyethylene, aluminum foil and paperboard. All together provides convenient, safe and lightweight packaging for the consumers. The outermost layer is polyethylene which consists of 15% which protects against moisture, gas and microorganisms to make the pack tight. The next layer is 80% of paper to provide stability, strength, stiffness and printing ability. The next layer is aluminum foil with 5% which protect product from air, light and off flavor. The two sides of aluminum foil is layered with polyethylene for adhesion purpose. In combination of all three materials provides excellent performance in the aseptic packaging of foods, which provides hygiene, safe and high nutritive foods. Aroma and freshness is preserved for long time. No need for refrigeration storage. Need not to add any preservatives. Low cost packaging material with minimum requirement. Now let me see about the types of Tetra Paks available in India. Tetra Brick Aseptic, Tetra Classic Aseptic, Tetra Fino Aseptic and Tetra Wedge Aseptic are the four different types of Tetra Paks available in India with various capacities and sizes with current legislative standards. This packaging is designed for consumer needs and convenience. Now let's see about commercially available aseptically processed foods. Dairy foods such as ultra high temperature sterilized milk, cream, fermented dairy products, condensed milk, flavored milk, buttermilk and beverages like fruit juice, carbonated soft drinks, vegetable soaps, soy tea, tender coconut water, tomato products, concentrates, puree, jam, baby foods, sauces, desserts, sandwiches and some of the commercially available aseptically Tetra foods. Now let's see the summary of this module. Ultra high temperature process done by direct and indirect methods. Ultra high temperature process distracts all the pathogenic microorganisms and spore forming bacteria. And the sterile product is packed with sterile packaging material in the sterile environment is known as aseptic processing. Three different types of packaging materials such as polyethylene, aluminum foil and paperboard are used in aseptic packaging technology. Aseptic processing and packaging is safe and hygiene quality packaging alternate. Many commercial products available in affordable cost, but the requirement of packaging material is increased in this packaging technology.